Thanks. I am really happy to be in this online conference and in this session. I am currently a postdoc at the Pasteur Institute in France, but I will present work that I actually did during my PhD at UCL. So I want to start by highlighting some of the different selective pressures that the first Native Americans encountered as they entered the continent, which included different dietary pressures, as well as new climatic conditions and novel environments. For example, the high altitude environments of the Andes that force many of these populations to adapt. And here I'm just showing some of the different genes that previous studies have found to be under selection in different Native American populations, affecting different phenotypes. But of course, perhaps one of the strongest selective pressures resulted from the different pathogens brought by Europeans after the colonization of the Americas. And again, here I'm showing some of the genes that previous studies have postulated as being beneficial against this selective pressure, most notably different HLA genes. And so the aim of this project was to study the genomes of modern Latin Americans to try to identify signals that resulted from these two selective pressures, that is, those encountered both before and after European contact. So what we did is to study samples from the CANDELA consortium, and which is a cohort that includes more than 6,000 individuals from 5 different Latin American countries, namely Mexico, Colombia, Peru, Chile, and Brazil. And for each individual, we had phenotypic and genetic SNP array data that we used to detect signals of selection. As most of you know, modern Latin Americans have ancestry stemming from 3 main components: a Native American, a European, and an African component, which we can see here in this global ancestry plot generated with admixture at k=3. And what I want to highlight here is how heterogeneous these different components are, both between and within populations. So for example, in the case of the Brazilian sample here on the left, we see that most of these individuals have high levels of European ancestry. Whereas in the case of the Peruvian sample here on the right, we see that most individuals have actually higher levels of Native American ancestry. But importantly, we also see that there is variability within the populations. So for example, here again in the Peruvian samples, we see individuals with different levels of Native American ancestry, which is something that we actually leverage in our method to establish when selection occurred. So here we have a schematic of our selection model. And what we do is model the allele frequency in a modern admixed population, here shown as a purple X population. And we model it as a linear combination of the allele frequencies in the 2 ancestral populations, K1 and K2, weighted by their respective ancestry contributions. And then what we do is we compare the observed, that is the present allele frequency, against this expected allele frequency. But deviations from this expectation can arise not only if selection happened after the admixture, that is along that purple branch, but also if it happened in any of the parental populations, in this case in the red or blue branches. And so what we do in order to differentiate between these two scenarios is that we compare the patterns of the expected allele frequency in the admixed population in relation to a given ancestral component. And so, for example, under neutrality, we see that the allele frequency of the admixed population follows the gray line, where individuals with no ancestry from K2 will have an allele frequency matching K1, and individuals with 100% ancestry from K2 will match the frequency of K2. In contrast, if there is selection in population K2, then individuals with 100% K2 ancestry will have a large shift away from the allele frequency of K2, here shown as a dashed blue line. And instead, if selection was post-admixture, then we see again a frequency shift from K1 and K2, but this shift is not nearly as correlated with the ancestry proportion from K2, here shown as a dotted purple line. And so what the method does is to compare the likelihoods of the data under these different models to classify each selected sites. And so before applying this method to our real data, we first evaluated the power of this method to detect selection, either post-admixture or in any of the ancestral populations. In this case, in Native Americans or Europeans only, as our 5 Latin American populations do not have high levels of African ancestry, as we saw in the admixture plot, and we would expect to have very low levels to detect selection in that source population. So we use simulation parameters that closely mimic each of our 5 Latin American populations, here shown in the rows. For example, using the same admixture proportions we observed and recent admixture times. And as expected, we see that power increases with higher selection coefficients, and that it also depends on how long selection occurred. The power also depends on the percentage of ancestry inherited from that source. For example, in the simulated Brazilian population, here shown in the upper middle plot, which has very little Native American ancestry, we see that we have virtually no power to detect selection in Native Americans. But overall, in many cases that realistically mimic our data, we see that we do have decent power to detect selection. And where we do detect selection, we typically classified it correctly as post-admixture, for example, the simulated scenario here in the left plot, or as selection in Native Americans, the simulated scenario in the middle plot. For small selection coefficients in particularly, we weren't always able to make a call linked to a high unclassified rate, but we typically obtain a high classification accuracy whenever we do make a call. And so, for example, on the right hand side, you can see we don't always get it right in some simulation scenarios. Again, particular with low selection coefficients, but for most cases designed to mimic our data, we do distinguish in these cases reliably. And so then we applied this selection method to the 5 admixed Latin American populations, and the results are shown here as a circular Manhattan plots, where each point represents a given tested site across the genome, with the significant selected sites highlighted in black. And we were able to identify some previously reported sites and genes. For example, here in chromosome 6, we were able to replicate the strong signal that our studies have reported at the HLA loci that we actually found in 4 out of our 5 Latin American populations. But we also identified some novel signals. For example, here in chromosome 10, we detected a strong signal only in the Peruvian samples, for example. And so overall, in our scan, we identified 98 candidate genomic regions, 54 of which contain at least one protein coding gene. And interestingly, many of our novel hits overlap with different genes implicated mainly in either immunity, pigmentation, or metabolic-related phenotypes. So then what we did in order to define adaptive variants, we basically check whether any of our significant selected sites overlap with previously reported EQTLs or genome-wide associated loci, basically to be able to establish whether these sites had a functional effect or not. And so to finish, I just want to highlight some of our strongest novel candidate genomic regions under selection. In this case, 3 examples where we identify signals of selection at immune-related genes. So here each plot is composed of 4 different panels, with the first one showing the selection scan, the second one showing SNPs associated to different white cell counts, the third one showing EQTL signals, and the fourth one showing the candidate genes of interest within the selected region. And so in the first plot here, we see a signal in the Chilean population. We see that the top SNP overlaps with the CD101 gene, which is a gene that plays a role in T cell activation. And we also see that one of the top SNPs actually controls the expression of the CD101 gene, particularly in CD4 T cells. In the second plot, again showing a selection signal in the Chilean population, in this case a signal that overlaps with the PTPN2 gene, a gene implicated in the development of the immune system. We again see that the top SNP is strongly associated to different types of white cell counts, and that nearby SNP also control the expression of this gene, but in this case particularly in natural killer cells. In these 2 cases, our model actually classified these 2 signals as being selected post-admixture in the Chilean population, but here on the last plot I'm showing a selection signal at an immune-related gene that the model classified as being selected in the Native American ancestors of the Peruvian population. In this case, the SNP was located close to the MIF gene, a gene that plays a role in the regulation of macrophage function. And again, we see that the stop SNP was strongly associated to white cell counts, and that it controlled the expression of this gene in different immune cells, but particularly again in CD4 T cells. We also identify a novel signal, in this case in the Mexican population, that overlap with the MC5R gene, which is a gene involved in pigmentation biology. This top selected site was actually in strong LD with a variant that is associated to skin pigmentation based on a GWAS that was conducted in the same CANDELA cohort. But the method was not able to confidently classify where the signal occurred in the Native American or European ancestors or in the Mexican population after the admixture. And lastly, regarding our novel signals and metabolic-related gene, here for example in the first plot, we identify a signal in the Chilean population that is located upstream of the CLC52A3 gene, which is a gene that has been implicated in the modulation of fatty acid metabolism. And interestingly, we observe that our top selected SNP actually regulates the expression of this gene in adipose tissue. We also identify two signals in the Peruvian sample, both located on chromosome 10, which were classified by our method as being selected in the Native American ancestors. The first signal here overlaps with a gene that has been associated with preeclampsia, which is a pregnancy condition. And this is particularly interesting because this condition actually shows high prevalence in many Andean communities. So it might be interesting to know the role of this selected variant and if it's indeed having an effect on the prevalence of this condition in Andean populations. And lastly, here on the third plot, we also identify another signal that overlapped the gene HKDC1, which is a gene involved in glucose homeostasis and has also been associated with type 2 diabetes. And in this case, we also found that the top select variant regulates the expression of this gene, particularly in muscle tissue and adipose tissue. And to conclude, here I presented a novel allele frequency-based method able to detect selection in an admixed population. The method is also able to determine whether selection occurred post-admixture or prior to admixture in any of the ancestral populations, and is also able to infer the selection coefficients of the selected sites, although this is a feature of the model that I didn't present here. I also didn't present all our novel candidate genes, but overall what we saw is that Native Americans seem to have been subjected to different selective pressures, whereas Latin Americans seem to have been subjected primarily to different immune-related selective pressures after the admixture. And with that, I would like to thank all the different co-authors as well as all our collaborators, in particular to my former supervisors, Garrett Hellenhall and Andres Ruiz Linares, to all CANDELA volunteers, and to you for listening.